High quality starting material, whether DNA or RNA, is always good to begin with in those next generation sequencing technologies. So in order to do that, we need really good protocols. Here is again we see the workflow for next generation sequencing, and we are actually talking about the extraction here. DNA and RNA, both of them, they needs to be in really good quality in order to be processed into the sequencers, and we have different protocols. Why we need that? Because even the best and most robust technologies, they only provide reliable results if our data is of good quality. There are some sensitive applications in NGS, so for that we obviously need that different contaminants, they must not be there in our materials. For example, like inhibitors, anticoagulants and enzymes, they must have been removed. Here is a famous DNA extraction kit. Normally it is mostly used, Kia AMP DNA mini kit. We can see different samples coming from different sources, and then we can have the starting material which we can get, and the total amount of nucleic acids, and then the DNA. For this NGS sequencing, even the small starting materials are also good, but it always good to have good amount of DNA. But they can work with even 2 to 10 micrograms of the nucleic acid as a starting material. So for example, if we look into the blood, if our starting material is 200 ml, microliters, we can have 4 to 12 nucleic acids each microgram, and total DNA what we get is 4 to 12 micrograms. So here in this chart, we can see different tissues and sources. So amongst them, for example, if we look into the lungs and if we look into the spleen, so these are the organs where we can have relatively less amount of DNA. We can start with less starting amount, and then we can get a fair amount of micrograms of DNA. If we start with cells which are the cell population of 10 raised to power 7, which is 10 million, we can get around 30 to 40 micrograms of DNA. So purification of high quality RNA can be challenging because RNA-easy is the standard technology which is used. Why it is challenging? Because RNA degrades in a quick amount of time. If we look into the extraction protocols for RNA, normally RNA is extracted in order to do expression studies, where we are interested in extraction of messenger RNA. So messenger RNA are mainly extracted by the protocols which are actually recognizing the messenger RNA based upon their poly A tails. So we have oligo DTs designed in these kits. So those DTs, they capture the RNA, messenger RNA that contains the poly A tails, which are missing in the other RNAs. So here is RNA extraction protocol in general. So we have different kinds of RNA. So whenever we extract RNAs, we have messenger RNA in the mixture, we can have transfer RNAs, and most of the time, or the major proportion we get is ribosomal RNA. So how to get rid of these other two RNAs and how to just have this messenger RNA? If you look into the structure, we see in the end we have an additional run of A's, which we call it as a poly A tail. And here in the extractions in our kits, we have techniques where we have designed some oligo DT matrices, so that have lots of T's. So since A binds with T, we can capture them, and then we can dilute rest of them and wash them away. So in this way we can only capture the messenger RNAs. But oligo DT methods which are actually capturing the messenger RNA based upon poly A tails, that has got some issues, especially if the RNA is degraded, or if that RNA has been fragmented. So then in most of the cases it will only capture the RNA from where the tails are added, from the 3' end site. So there are alternative protocols that claim to be work best with if we have degraded RNAs or if we have fragmented RNAs. So we conclude that high quality starting material is essential for sequencing and accurate data analysis. And if we look into the RNA extraction protocols, we see that oligo-DT based methods, they have some issues.